vocabulary words that I would really like to go over with you that will help you understand the section, especially the first part of chapter 14 and the all of it. Chapter 14 is going to be divided into probably 4 lectures. So today, tomorrow, Thursday, Friday, today, tomorrow, Wednesday, Thursday. Friday, I'm going to post an invitation for a Zoom meeting so that we can spend some time going over the problems, or you guys can ask questions if you have questions about particular of the genetics problems, because that's a big part of chapter 14 is learning how to read a genetics problem and put it into a Punnett square and solve it for the answer. So in order for us to get a little bit of a better handle on this whole topic. So chapter 14 is called Mendel and the Gene Idea. All right, and it is Mendel lived and worked, and the only reason I'm telling you this is because it has a lot to do with what he could see. He was born in 1822, and I believe he did most of his experiments in 1857-ish. So he lived in Austria, what was Austria then, but what is now the Czech Republic. And the reason this date is important in the context of the history of science is for a couple of reasons. First of all, you need to know that microscopes were mostly single lens, simple microscopes or compound microscopes that weren't super high resolution until about the mid-1850s, mid-1800s. So I'm just going to put 1850s. Also, this is the time with better microscopes, and some of you may have learned these 2 guys before. These 2 guys, Schleiden and Schwann. Their names aren't particularly important, but they're the 2 people, Schwann with, I believe, plant cells and Schleiden with animal cells, that described cells as these small living units of life. So there's a lot of important advances, documentation, a lot of important things going on in the 1850s. With these better microscopes, not only can you see cells, but you can also start a little bit later, people, after Mendel's finished, people are going to start to be able to document the stages of mitosis and meiosis. And the other last important date person at this time, I want you to just remember back to chapter 1. I guess I should use a different color. Let's use yellow. Darwin is going to be publishing his book, Evolution by Natural Selection, and that gets published in 1859. So right around this time, okay? So there's a lot of big stuff sort of going on in science all at the same time, a lot of ideas all coming and swirling around together that basically kind of help Mendel put things together that maybe somebody else had already thought of but hadn't documented really well. One of the challenges with chapter 14 is that it's, again, a whole new set of vocabulary. And if you don't know the vocabulary, you don't know the words, it's really difficult to follow along of the problems and all of the stuff. So I'm going to make a list of vocabulary words here and then try to talk to you about, I made a list so I won't forget any of them, and then try to talk to you about them one at a time. So I'm going to put down genome and then chromosome and then gene, which goes by many names, characteristic. Sometimes that gets shortened to character / trait, although trait is kind of not in favor right now. But you'll hear me say it because that's the way I learned it, and that's still the way some of us talk about the characteristics. Allele, that's a funny word. Mendel didn't call them alleles, he called them elements. Well, he used German, so he called them, I don't know what he called them in German. And then these two words, phenotype and genotype, those are more modern words. True breeders, which we also call the P generation or the parent generation. The F1 generation, which stands for first filial generation. That's what F1 stands for. Homozygous and heterozygous. Let's see how many words is that? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Oh yeah, and dominant and recessive. So it's 12 words. And this should really say dominant allele and recessive allele. These 12 words, you must memorize a definition for them as soon as possible. I know, you're studying for the test for tomorrow, right? I get it. But as soon as that's over, tomorrow night, or tomorrow before you take the test, sometime you have to learn these words because the rest of chapter 14 won't make any sense unless you know these words and their definitions, and you need them to be able to solve the genetics problems that we're going to talk about tomorrow, and then Wednesday, and then Thursday, and again on Friday. So I was going to start here with genome, chromosome, gene first. So I'm just going to add another whiteboard. So the genome of an organism, that is all the genetic material in an organism. Right? And we know that the genetic material is composed of DNA, deoxyribonucleic acid. So it's a type of nucleic acid that we learned in chapter 5. Right? And we have to say all the genetic material because most organisms have their genetic material arranged in chromosomes. Plus, sometimes they have some extra bits of DNA. So, like in you, for example, an extra bit that I can think of off the top of my head is I'm going to draw your friend the mitochondria. I know you were hoping you'd never have to see this again. I know. Here he is. Right? And you might remember that when we talked about the mitochondria, we said it had its own ribosomes, right? And its own chromosomes, so its own little bit of DNA, right? Another type of DNA, especially if you're a bacteria that might be present, you might have something called a plasmid. And as an animal cell, you have some incorporated viruses probably into some of your DNA and some transposable elements that sometimes move from one place in your DNA to another place in your DNA. So the genome is all the genetic information that you have, right? All in an organism, like at any one time. Now, this is arranged in chromosomes. So I'm going to go back to my other word here. Get rid of my mitochondria. Right. So back to this, chromosomes. And again, if you're a prokaryote, if you're bacteria, right? I'll make a round bacteria. You usually have, as I just drew in the mitochondria, you usually have a single chromosome that is double-stranded, double helix in a circle. It's usually circular. And if you are a eukaryote, right, you, of course, would have all of your chromosomes are all inside your nucleus. There's the dark spot, there's the nucleolus, and I'll make them blue, right? And we saw in both chapter 12 and 13 when we're in G1, when the cell's just doing its regular life, I'll draw mitochondria in there and some endoplasmic reticulum to make some proteins, Golgi, right? Some ribosomes. Okay. So these chromosomes most of the time are relaxed, right? Fairly relaxed, and they aren't condensed into individual chromosomes that we can see, but we know that if we look at mitosis or meiosis, we can actually see the individual chromosomes. And remember, we looked at that karyotype that showed all the chromosomes from a human, right? So if we were talking about, so this would kind of be like an animal cell here. If we were talking about a plant cell, those are usually kind of square shaped. A lot of them are very square shaped. And if we were talking about our onion cell in particular, right, we would see 3 chromosomes. We would have chromosome 1, right, that's been duplicated before meiosis. Well, let's not do that. Let's just do this. Let's just say, so we would have chromosome 1 and chromosome 1. I'm going to make it with diagonal lines in it. Remember when I had the pieces of the clothesline, I had pink ones and white ones, right? And then our chromosome 2 is going to be smaller, and that one too would be stripy or plain. And then our chromosome 3 would be even smaller than that, and it also would be stripy or plain. So I'm going to make this one stripy. Kind of hard to do, right? And of course, this would really all be inside the nucleus, which I didn't draw very well here. So let's make this the nucleus. And I'm just going to cheat and try to make my plant cell look bigger here. There. So that's in the nucleus inside the plant cell, which of course would have chloroplasts, stacks of granum in it out here, right? And mitochondria so that it can make ATP. And of course, a big vacuole, right, full of water, wherever this is supposed to be placed. So the reason I'm emphasizing the plant cell is because we're going to talk about characteristics in peas in particular, because Mendel studied pea plants. And so I wanted to highlight the plant cell one more time, okay? And in the peas, in particular, in the plants that Mendel studied, there were 7 chromosomes. So N would equal 7 for the pea plants, or 2N would be 14, 2 copies of each chromosome. Just like in my cell here, where N equals 3, I have 6 total chromosomes, right? 2 copies of each. Okay, now there's one more thing we need to say about chromosomes. They are composed of 2 things, DNA plus proteins. Particularly in the eukaryote. There's some proteins in prokaryotes, but not so much. Those are mostly just DNA. But in eukaryotes, it's DNA and proteins. Okay, and some people will even go as far as to make you remember they're a special kind of protein that we call histone proteins. That's going to come up again in like chapter 16 or 17. So I just throw it out there to help you remember. Now, the tricky bit is, so genome, chromosomes, I'm going to erase the genome part up here so I can draw a little more here. Let's just look at these green chromosomes here from this plant cell. If I were to draw them much larger like this. Right, and I'm going to call this one chromosome 1. That's kind of a mutated one. Let's try to make that look a little better. Still not so good. There we go. Okay, so 2 copies of chromosome 1. Now, This brings us to our next word, I think, on our list, allele here. Okay, or a gene, actually. Let's focus on it being a gene. All the areas of this chromosome are pieces of DNA, and they're matching areas on the two chromosomes that are the same number. And like right where I've drawn that red horizontal lines, that band, if you think back to the karyotypes that we looked at the other day, that is a place. A gene is a location, a particular spot, literally a location, and they have numbers and they have letter designations. It's a location on a chromosome. You can think of it like as a feature of an organism or a characteristic. Or a trait or a feature. Okay, so in peas, which is what we're going to be talking about in chapter 14, one of the chromosomes in peas, one of those 7, has a particular gene, a location for flower color. And I don't know if your textbook goes as far as telling us which chromosome flower color is on. Take a quick peek. It does not, but let's just say it's on chromosome 1, right? And you could isolate it right here to that spot. That's where you find the DNA that tells the flowers to make a protein. And in peas, we're going to see that the flower can actually be either purple flowers or, not an and, or white flowers. So each plant is going to carry genes on chromosomes inside their nucleus to produce either purple flowers or white flowers on the plant. Now, we refer to those two different colors, those two different versions, purple or white. That's the definition of an allele. So an allele, or an element, like I said, as Mendel called them, an allele is a version, or sometimes people will say a type, or I like to think of it version, a type, a kind, flavor, right, of a gene. And there are often two, right? 2 alleles for flower color. Purple alleles, white alleles. How the organism looks depends on which versions of these chromosomes they inherit from their parent as a result of our old friend meiosis. Because, right, when this particular plant cell that I've drawn here undergoes meiosis, meiosis 1, meiosis 2, right, we're going to end up with 4 plant cells that are gametes, right? And each one of them can only get one of those chromosome 1s, either the stripy one or the plain one, right? So then it's going to matter what the genes were right there, if it was the gene for purple or the gene for white. Because depending on then what fertilizes any of these gametes, that's how we'll get back to 2N. That's going to give us plants that had either purple flowers or white flowers. But each offspring only inherits one copy of this chromosome from a single parent and the other copy from the other parent. Trying to tie that back a little bit to meiosis. Okay, what's my next word? Let's do phenotype next. And then we'll talk a little bit about Mendel's experiments. So let's see. I should probably just take another board. I don't want to do that because, oh, there we go. Okay, phenotype. Phenotype is the appearance of the organism, right? With respect to a characteristic, right? So in this case, your book's going to talk about flower color, so I'm sticking with that on purpose and not trying to introduce another example that's different from this, so that when you read your book, it's the same as the examples that I gave in the notes here. So phenotype is what the organism looks like. Usually people say that. They say, what does it look like, right? When we start doing the problems, and I'll say, okay, what color is it? Or what does that one look like? Another way to think of it is it's the physical appearance. And I think that's helpful because physical and phenotype both begin with pH. That's what helps you remember. That's what it looks like. It's what it physically looks like. Are the flowers purple or are they white? Okay, what's our next word? Okay, now we can talk a little bit about Mendel's experiments and how he set them up. And that's going to lead us to talk about some of these other words. So I'm going to go back to this board and let's talk about true breeders, also known as the parent generation. And this is not in the casual way that anybody is a parent, right? Or any organism is a parent. It's a very specific meaning of parent. And we shorten it to P generation, like that. Okay, so we only call the parent generation or the P generation. We only refer to them as true breeders. And what that means is in Mendel's experiments, for example, with the Ps, it means to be a parent generation, you have to only express one of the phenotypes for a trait. So in our case, for example, a true breeding P for flower color, we would have plants that only ever had purple flowers. How do you do that? Well, that's the little bit crazy part about what Mendel did. That's in peas, he bred the pea plants for 7 generations to be sure that every pea plant that had purple flowers only gave offspring that had purple flowers. The other parent, in this case, the other parent organism in this particular example that I'm giving you would be a parent flower plant that only for 7 generations gave white flowers. After he knew that he had true breeders, or sometimes in middle school and high school, they'll call them pure breeders or pure lines, and then they'll call the F1 generation hybrids. Our book doesn't use that terminology. That's like a simpler terminology. So we're going to refer to them as true breeders and as the F1 generation. So after he bred the plants for 7 generations, then he took a true breeding purple flower plant and he crossed it with a true breeding white flowered plants. And I'm going to label this P generation because it was true breeding parents. And he called, if he crossed the flowers, which means he removed pollen from one and put it onto the purple flower, and then he removed purple pollen and put it onto the white flower, and went through this really complicated protocol, which you can see in your book. And then waited for them to form seeds, and then planted the seeds and waited for those seeds to grow into plants, and then looked at the flowers. What he found was in his first generation, his F1 generation, all the plants had purple flowers. And this was, he had a working hypothesis that the current theory about inheritance was that traits were blended together. But Mendel didn't think that was true, and he thought that this gave evidence to that because if they were blended, he thought he was expecting to have purple flowers with white stripes, or maybe a lighter purple, but they were all purple and they were all the same purple. And so he thought, maybe things aren't blended together. Maybe those, whatever carries the trait for white, maybe it just disappears, right? However, when he let the F1 plants self-fertilize, because flowers, angiosperms, have both male and female gametophytes and gametes inside of them, he let them self-fertilize, they're gonna be, you put little bags on them, let them be fertilized by the wind, by the water, wait for them to form seeds, plant the seeds, wait for this plants to flower. What he saw in the next generation, which he called the F2 generation, he saw some of his plants had purple flowers and some of them had white. But what's important is how many. 3/4 had purple flowers and only 1/4 had white flowers. And if you look in your book, you'll see that he counted, this represents a count of over 705 plants with purple flowers compared to 224 plants. So it's not just like he grew 12 peas and was like, okay, this is a thing. Repeated it many times and obviously counted lots and lots of plants and their flowers. After he completed this, he added some, we can add some more things to our vocabulary list. So we've talked about true breeders, F1 generation, but now we can talk about these two words here, dominant allele and recessive allele. So Mendel formulated a principle, he called it, and now we call it a law because it's been proven and demonstrated over and over again with many, many plants and many, many animals and many, many fungus and many, many bacteria. And the law is called, it's his first law, it's called the law of segregation, or the principle of segregation. And it states that some of the alleles, we call them dominant, and a dominant allele is the allele that is expressed by the phenotype of the F1 generation. I know that's a lot of words that we just learned, right? So in this case, let's keep it simple. In this case, the dominant allele for flower color is going to be purple. And it's usually, but not always, the darker color, or I shouldn't say that, but a lot of times it is, especially in plants. The recessive allele, this is the allele, we say, that is hidden or masked in the F1 generation, but reappears in the F2. So this is going to be an important thing for us to remember when we try to start doing genetics problems, because sometimes the problem is going to identify the dominant allele and the recessive allele, or sometimes they're going to just describe two true breeders being crossed together, and they're going to give you the phenotype. They're going to tell you what the F1 generation looks like, and then you have to use that bit of information to determine yourself, which is the dominant allele, which is the recessive allele. I'm going to go back to my list. All right, a couple more words here. We're almost done. Homozygous and heterozygous. This has to do with a particular individual, and I'm going to go back to this drawing, talk about that. Maybe this one even, actually. So if an individual is homozygous for a trait or characteristic, that means they have two copies of the same allele. Same homo, same, right? Same allele. So in our case, they would have two copies of the purple allele or two copies of the white allele for flower color. Right? So if you're thinking about the chromosomes and the genes that are on the chromosomes, again, right? We don't know here if this is purple or white, but if it was a homozygous individual, both of these areas for flower color would be purple, as in the example that I just gave, right? If an individual is heterozygous, oops, then, as you might suspect, that means the opposite. That means the individual has one copy of each of the alleles that are in the gene pool for a particular trait. Right, so heterozygous, because diploid organisms are 2N, so you have that equals two copies, right, of genes for a trait or characteristic. So in the case of being heterozygous, you have one copy of the purple allele and one copy of the white allele. Right, so hetero, one of each. And again, those are words that you're going to need to be able to call up and remember what they mean in order to solve the genetics problems. So if we look back to what I described here, right, true breeders, the true breeder or the P generation are always homozygous. It's not very good writing there. There we go. Try again. Always homozygous. Two copies of the same for purple, two copies of the same allele for white. The F1 generation, I should make this red. The F1 generation is always heterozygous because they're a blend, you know, they're a one, they got one gene from each parent. F1 is always heterozygous. Parent generation is always homozygous. Okay, I think I'm going to stop there. Okay, I need to do that. How do I get back? That's it. Okay. Questions anybody has about this that I've just gone over, these definitions? I'm gonna check it again. Yes, no. These words are all defined in that in the same video in the part 1 of my chapter 14 video. as well, probably in a pretty similar manner. I think I was using the chalkboard that day, and I went through all the definitions, and I stopped short of actually writing out the complete law of segregation because I was using the PowerPoints and the chalkboard and writing down all these definitions, and it was a little overwhelming, and it is a little overwhelming. The law of segregation itself, Mendel's first law, has 4 parts, and so I did add them to the note outline, and I'm going to write them out for you here. Okay, so Mendel's first law. All right, it's called principle. Sometimes you'll see it called principle, sometimes you'll see it called law of segregation. And 4 parts. Okay, the first part is that there are alternative forms of alleles. So like we were just talking about, there are genes for purple, genes for white, and these alternative forms are important for variation, or account for the variation in organisms. Second, if the organism is diploid, in a diploid organism, the zygote inherits 2 copies of a gene or 2 copies of a chromosome, 1 from each parent. And that's important to remember, 1 from each parent. Three, if that zygote, and this is kind of belaboring the point a little bit, but it's how your book does it, so that's why we're doing it that way. If the zygote inherits 2 different alleles, they would be heterozygous, right? And remember, the dominant allele would show in the phenotype. The recessive allele would be hidden or masked. And then 4th, this is where we actually get to the principle of segregation part, the 2 alleles for a trait, I'll put for character, will separate or segregate from each other during gamete formation. AKA meiosis, right? And this is a big important and. There is an equal chance that either will end up in any of the gametes. I'll say any gamete because I'm kind of out of space here. And that's important because there is an equal chance that if the organism is heterozygous, there's an equal chance that they're going to produce gametes. Half their gametes will have the gene for purple, and half of their gametes will have the gene or chromosome for white. Okay, so that's the end of what I